multipurpose vectors for use in yeast. Special purpose vectors have been developed that can incorporate or contain the properties that are desirable for bacteria or E. coli, or in other words, we can say that we try to incorporate all the beneficial properties that are present within the bacterial vectors or plasmid. Now, in case of yeast, some plasmids like that contain F1 origin that can later on help the target inserts for sequencing, or that can produce the purified fusion proteins. They have been preferred as a specialized vectors. But when we consider different vectors for use in yeast, then we must also take into account that how the target proteins that have been expressed within the yeast cells, how they are going to be synthesized, and after synthesis, how they will be transported. So all these parameters we have to take into our mind. So for this purpose, we have to use special purpose vectors that can also mediate the transportation of the foreign proteins. For example, in case of yeast, if protein it is being synthesized, then after synthesis it is being transported to endoplasmic reticulum where it is being processed, and after endoplasmic reticulum, then this protein it is further transported to Golgi apparatus. So the vesicles from the Golgi apparatus, they are being transported to the membrane so that vesicle can be fused, and protein they may be transported on the membrane or in the periplasmic space or outside the cell. But for yeast, mainly two types of proteins they are mainly transported outside the cell, like some mating factor pheromones that is commonly called as the alpha factor. It is transported outside the cell, or other genes that are involved in the synthesis of toxins. So the toxins are killer toxins, simply we can call them. They are transported outside the cell. Other proteins, they remain within the cytoplasm or within the periplasmic space. तो इस सिचुएशन में जो हमारा एक्सप्रेस प्रोडक्ट है उसकी पॉसिबिलिटी है कि वो मीडियम जहाँ पे हमने यीस्ट को कल्टीवेट करवाया है वो वहाँ से सेल के अंदर से मीडियम में रिलीज ही ना हो. तो इस पर्पस के लिए हमें इसको एक फ्यूजन प्रोटीन के तौर पर एक्सप्रेस करवाना पड़ेगा. लाइक आवर टारगेट प्रोटीन, it can be expressed as a target protein with alpha factor. तो इस सिचुएशन में नॉर्मल प्रोसेस होता है कि जब हमारी फ्यूजन प्रोटीन सिंथेसाइज होती है तो वो ट्रांसपोर्ट होती है टुवर्ड्स द एंडोप्लाज्मिक रेटिकुलम, एंड then it is processed there and then transported to Golgi apparatus, and within the Golgi apparatus then its signal sequence that may be hydrophobic region, maybe 20 amino acid, it is being removed, and then those vesicles they are transported outside the cell. So while considering the use of different plasmids, we should take into account that those plasmids are vectors we should use that can also facilitate the release or secretion of proteins outside the yeast cell.